My lecture was based on my work over the last 20 to 25 years, where I've been working with the morphine-like drugs. I've always been interested in old drugs. We have a lot of medicines on the market which are very old, they may be 50 or 60 years old, and these old drugs haven't been tested to the same requirements that a new drug needs to be tested these days. So a lot of the basic pharmacology we still don't have a good understanding of. Why is it that one drug like morphine will work in some patients, it doesn't work in others? You give them another opioid and it'll work in the ones where morphine didn't work, whereas their basic mechanisms appear to be the same. The other thing is I'm interested in these drugs because the way the body breaks them down produces other opioids which have activity in terms of pain relief and in terms with adverse effects as well. So if you're going to be looking at these drugs in vivo, whether it's in an animal or a human, you've got to be thinking about the other products that the body is making from them, their metabolism. That means you've got to be able to identify these metabolites and test them and even make them. So, that's a long and hard process. But nobody else works in this area, so to create your own niche, that's the sort of thing you have to do. The key points from the lecture were that these opioids, not only do they bind to a specific receptor, we call them mu receptor, and that then affects the signal to reduce pain up into the higher centres of the brain. So we've known about that for a long time. But some work we've done over the last 3 to 4 years, we've actually shown that there's a second target where these drugs work. And this target has an opposite effect. And the target is called a toll-like receptor. And these receptors, one called TLR4, it is not really on neurons, it's on microglia. And microglia were often thought of as just the glue holding the neurons in our brain together. But we now know that they have all these receptors. Within the cell itself, they can manufacture all sorts of things. So drugs like morphine actually bind to one of these toll-like receptors. And within the cell, it makes a cytokine called interleukin-1 beta. And interleukin-1 beta is a pain-producing chemical. So morphine is also doing that. So on the one hand, it's relieving our pain, but on the other hand, it has a component of producing pain. And we think that's one of the reasons why there's this enormous variability in how much dosage of morphine people with cancer pain or after surgery require. You've got the good effects and you've got these bad effects. And some people might have more of the good effects and some people might have more of the bad effects, saying that they need more morphine. So it's a completely new area of drug discovery, these what we call toll-like receptors. The other important thing is, particularly in humans, many of these opioids are broken down in the liver by specific enzymes to products which are active. And the enzyme, the gene for that enzyme is polymorphic. And what I mean by that is that for one of these genes, about 10% of Caucasians, that gene doesn't work and they don't produce the metabolite, which is the major active pain-relieving substance. So for example, codeine, from some of the work we did 25 years ago, showed that 10% of people didn't form morphine. And we think that morphine is what causes the pain relief in people who take codeine. In the UK, there are 750 million tablets of codeine that you can buy over the counter that is consumed each year. And we still don't know exactly today whether 10% of us don't get any pain relief, so it's a useless useless drug. So we were trying to think about how we could design a study to actually prove that. Somebody asked me about cannabinoids, because cannabinoids by themselves don't do a lot in terms of pain relief. But if you give them with a drug like morphine, you get more pain relief than morphine by itself. So there's some sort of interaction there. These cannabinoids are switching off the pain-producing effects like a drug like morphine. So you only then have the pain-relieving effects, and that's much better because the pain-producing effects have been dampened down by the cannabinoids. So there's the possibility of having a collaboration with one group here to look at this in more detail.